All right, it's great to be back here at Ferment, and today we want to share a little bit of our journey of the project we've been doing with Ginkgo, which has really been designed to look at detecting the immunomodulatory activity of a large panel of gut microbiome-derived bacterial strains that we've identified from our proprietary databank. Now, when we think about microbiome therapeutics, there's really two aspects that I want to highlight. One, I think we'll all agree that the science is unequivocal on the ability of the gut microbiome to essentially be a sensor, a modulator, and a translator of key biological signals. However, our ability to identify the source of these signals and then harness them as precision therapeutics has really lagged. And at Microba, we think it's lagged due to three primary problems. One, deficient analysis and fragmented data sets. Two, if you look at the bioinformatic tools, they're riddled with errors. And three, generally underpowered data sets to try and make the correlations that we need to turn these things into products. So the way that we've addressed this is to think about a very large-scale combination of host data and microbiome metagenomics with deep neural network analysis to really identify the biological dark matter, the things that other people can't see, and then turn that into the products of tomorrow. So what's been super exciting in our program with Ginkgo is that we've been able to probe our proprietary data set and extensive microbial biobank to drive initial lead identification of around 200 strains. And importantly, that was down-selected from around 60,000 genomes, and we used a bioinformatic rationalization to identify these. We've then worked with Ginkgo to design both a high-throughput growth system and then a deep bioanalytical cascade to probe the biology that sits underneath that. We're about 2 years into our program, and we're now super excited to finally narrow down on those lead organisms that we're going to take forward. I also wanted to share with you, what does this actually look like? How far across the microbiome are actually going? So if we look at our full 200 strains, you can see that it spans the complete phylogenetic space of the microbiome. If we narrow down to around the 30 or 40 that we've selected now, we're still seeing a huge amount of diversity. And typically, these are things that no one else has seen before. They've never been cultured before. These are really novel bacteria. But I guess I just want to finish with showing what have we actually seen in the data? What's been the snapshot? Well, we've seen an incredible dynamic range of the ability to modulate the immune system. We're talking about inflammasome activity. We're talking about Th1-driven activity. And then we've been able to actually do that down selection looking at a raft of measures, human metadata and disease association, assay potency and biological activity, and finally, in silico safety and manufacturability. So with that, I'll wrap up and just say in summary, this approach has been super exciting for us to deploy large-scale drug discovery program for autoimmune disease on an unparalleled number of unique microbiome-derived strains. Thanks very much.